Hello, everyone. Welcome back to the Gut Feelings podcast. Today we have Dr. Yasmin Mohseni with us today. She is an immunologist working at biotech in cancer and immunotherapy. Dr. Yas, welcome to the podcast. We're happy to have you.
Thank you for having me.
We have a lot of questions today, but one thing that I really wanted to start off with was a recent Instagram post that you had. And for those listening or who may not follow Dr. Yaz on Instagram, is that you were recently diagnosed with IBD last year.
Yes.
And your post, the one that I really liked, it really spoke to me. I'm just going to read it now. I got my diagnosis in February last year. Despite being confident in biologics, I'm embarrassed to admit to myself that I was terrified of starting. I felt stubborn and thought I can do this myself. Flip. I'm an immunologist, for heaven's sake. So I understood what it's like to be a patient who is desperate to try and heal herself the natural route. Despite being a scientist devoting her life to inventing drugs that help the immune system like these. Feels a bit ironic, doesn't it? Yeah, I relate 100%. You go on to mention a few things that you adjusted to help out your body in general. And then the post continues to read on. Did I feel like a failure that the immunologist wasn't able to cure herself naturally? No, because WTF, because like your immune cells have been evolving over 1 billion years, and you are not suddenly going to reset by curing your childhood trauma or fasting. But do I feel better and happier and healthier? Yes, I do. So I just wanted to say thank you for sharing something like that, because Ashley and I, we talk about this too with both of us having IBD, being dietitians and working in the IBD space, that it's like that kind of thought that it's like, oh, you're a dietitian or you're a scientist, like you should be able to figure this out. But that's just not how it works.
No. And I don't even think it's more that. I think it's also more like, hey, actually, we get it. We're not just doctors, scientists, like professionals in our own little echo chamber just trying to pump you with drugs. We actually understand the emotions, like the fear, the sort of the guilt, the shame, the self-blaming. We get all of that, as right, as wrong as all of it is, but we understand what it's like to go through that process. So I think that sort of was something that I really wanted to bring across, because at least with my social media, I was always labeled, because of the nature of like my content initially, and just being a scientist in biotech, which, right now my biotech company is still in that startup phase, but eventually there's only two routes. Well, there's three. We don't have to talk about the third, but the third is, it just fails. But it's just, a lot of startups do fail. But the other two options are you get bought out by pharma, or then you go public and essentially by yourself try to become a farmer, per se. It's a little bit more complex than that. But I was always getting these accusations of, I just work in pharma, so I just want to pump people with drugs because all the money I'm making. And it's like, no, it's really not. No. And also, I know what it's like to be on that end. And it's interesting because I was firmly believing always in biologics. I firmly believe in all the amazing breakthroughs in the technology to be able to deal with all of these various diseases or various immune pathology. I mean, these days, so many diseases are linked to the immune system. It's just crazy how much we've grown to understand. But when it suddenly becomes you and your body, it just definitely gives you that different lens. So yeah, I didn't realize that both of you also have IBD. So I knew one of you did, I think Ashley, but yeah, I guess it sort of spurs us on even more, doesn't it?
Yeah, it's so accurate. And I feel like it's so vulnerable and human to, I think everyone on our team has gone through this same thing of like acknowledging being the patient and making space for this, having this understanding of how something works and the value of it and speaking to it, but also struggling with the vulnerability of being a patient and having to make those choices and recognizing the long term. It really does make you realize that no matter where you are in life, like as a patient, you go through these phases, so to speak, of like, it being so long term too. I think there's the chronic nature of it. Like I was telling Danielle even a couple weeks ago how I was struggling with, the long-term aspect of it and just not really wanting to be on meds, but also understanding that I wouldn't guide someone down that path of like, don't be on meds. So yeah. So thank you for sharing that. It really spoke to both of us, just knowing that we're not alone. Like there are other people speaking the same thing that we struggle with. So
Yeah.
I'd love to know too, like everybody has that starting point or that initial story of how they got into the path they're on, and being a scientist and immunologist, maybe you can share a little bit about what that journey was like to you and how you got here.
Yeah. So, if it's not obvious from my training, from my accent, my training was in the UK. I am British. I'm from London originally. So I started off with doing a biochemistry degree at the University of Warwick. And I wasn't really sure whether I wanted to go down the hardcore bio route or the hardcore chem route, medical school. And I sort of thought to myself, well, maybe let me just do a master's just to like buy myself an extra year to figure out what I want to do, essentially. And I was always quite fascinated with immunology. And that was actually my optional lectures. Those weren't my core, but I just picked one immunology module in my second year and one in my third year. And I think what fascinated me was it seemed like these cells actually had conversations with each other. They actually communicated with each other. And it's each immune cell had a job, a very specific job to do. And this one goes and recruits this guy, wakes up this guy, switches off this guy. And I just thought, this is really cool, but weird. It's just the idea that these cells are almost humans in a way. So I was very curious about that. So I went on then to do my master's in immunology at Imperial College London. And that was a really great experience. I was in an asthma and allergy lab for a year for my master's degree. And then once I graduated, I got to stay on for an extra year at the lab as well. But when I was picking PhD programs, at this point, I still didn't know what I wanted to do. But I was so interested by the immune system that I thought, let me just go do a PhD because I wanted to know more. And, I think that's always a good sign. If you don't know what you want to do, but something really interests you and you just need to know more about it. And yeah, of course, then that paved the way for my career as an immunologist. But I was looking at PhD programs, and asthma and allergy really interested me. But then there was this position at this lab that I really looked up to the professors at King's College London, and it was looking at organ transplant rejection. So it was looking at, specifically in this context, would have been liver transplant rejection. I was also exploring heart transplant rejections in kids, but it was using this technology that it's been FDA approved for cancer, and it's relatively novel, as in gone into patient around like 2010. And it's just been booming like the liquid cancer space, like the blood cancers. So it's applying that same technology, but doing the opposite. Instead of killing something, it's tolerizing, it's switching to try and make your immune system accept something. So that can be applied to both an organ transplant, but also autoimmunity. So the core cells that I specialized in were something called Tregs, or regulatory T cells, which I'm sure you guys as dietitians have definitely, in the immunology space at least, you've been hearing a lot about Tregs, hopefully. I'm biased. I think they're the most important cells in the immune system. Very biased though. But essentially their job, their function is to suppress inflammation. It's just to keep inflammation at bay. So they call off the attack essentially, and their job helps to prevent autoimmune diseases in your body and just an overactive immune system. So the idea is, okay, we know that their job is to suppress, to calm down inflammation and just to make everything chill out a little bit. Can you engineer them in such a way to drive them to an organ transplant? Because with organ transplantation, it pretty much all the time there is rejection. That always happens. Even as much as we try and match as close as possible donor to recipient. And, some surgeons in the space might challenge me a little bit on that, because there's definitely spontaneous acceptance of organ transplants. There's definitely more organ transplants that are a lot more problematic in terms of rejection than others. But in general, patients will have to go on immunosuppressive medication. And the challenge with these kinds of immunosuppressive medications for transplantation is they're very hardcore. So not only do they not fully suppress the immune system, which is a good thing because you kind of need an immune system. So immune cells will still come and take jabs when they can at the organ transplant. but also they're quite hardcore on the kidneys as well, and just some toxicities. So the goal is to be able to wean patients off of immunosuppressants. So that was what my PhD project focused on, is that can we use this technology that we're using for cancer just to engineer the Tregs to go and basically stand guard and protect the liver transplant. So yeah, that was my PhD project. It was 4 years, and then I got to stay at a startup in London, which was founded by my PI and 5 others. It's called Quell Therapeutics, and pretty much looking at the same thing. Can we now at least, take this into patients? And they're doing very, very well, which is pretty awesome. And I miss them all. They were like family to me. But then I had this opportunity to come to the States. And I sort of got to that age where it really felt like now or never. I didn't have anything massively tying me down to the UK. Of course, my parents were there and my friends were there, but I really needed a new adventure, and it was California. So I accepted a position in the US and I came out here 3 years ago. And I'm now working at a cancer biotech company in Thousand Oaks, Agora Hills. And yeah, so I've segued into cancer, which maybe was inevitable, but my heart is definitely in autoimmune diseases. So I think at some point I will segue back into that space. So it's a bit of a ramble as to how I got to where I am now. Hopefully it makes more sense though.
Yeah, I was like captivated with how you described the immune system. So I know a few scientists, and I know that they're not all great at really making it interesting like you just did. But yeah, like how you described it makes the body so much more interesting when you can really understand it. Like it's down to communication. Yeah, just interesting. I almost forgot my next question because I was so wrapped up in what you're saying. But that's also what I find, what kind of drew me to gut health and microbiome, understanding that, just because it's so fascinating to think about when we eat food that we're feeding these tiny gut microbes in our gut, and that's producing all these side effects. But yeah,
I think that's an interesting point though, because I think for so long the hallmark of the immune system, one of the core hallmarks, which I still kind of stand by, is recognizing self to non-self, like what is you and what is not you and what needs to get out. However, we know food is not self, and we know the microbiome is symbiotic. It's not self, but it's like symbiotic to us. We have this friend relationship. So the immune system, the fact that it's evolved in this way to be like, oh, you are a friend, you are to the microbiome, and you are not harmful to food. But of course, we know sometimes this goes wrong, like autoimmunity and in allergies. So that's why it's so interesting, because something has gone wrong. And how do you restore that balance? Because ultimately, I think, again, if you talk to immunologists, we all have different lenses of what the hallmark of the immune system is. But I think the immune system always tries to get back to balance. And if you tinker with it, which is interesting, being in the immunotherapy space where we are trying to, I guess, manipulate or engineer the immune system to kind of be better at something, like better at fighting cancer. Immune checkpoint inhibitors won the Nobel Prize, for example, for revolutionizing the cancer space. And it's just basically helping your immune system out to fight off the cancer, because that's what part of your immune system's job is, to clear off cancer as well. So, but if you tinker with it too much, then if you get too much, that's why the concept of immune boosting is so fascinating to me, because you don't really want to boost your immune system, because if you boost it, it can kill you. We know from food allergies, from anaphylaxis, or like venom allergy, anaphylaxis, that can kill you within minutes. And that's your immune system that does that. So do you really want to boost it too far? Like you want to boost it to clear off cancer, but you don't want to also have all that toxicity. So yeah, it's just very interesting. It's constantly blowing my mind, the new things that we're learning about the immune system.
yeah, for sure. So I guess walk us through, what's a day in your life, in your role, and even outside of your role, like on Instagram, or what does your day look like?
Yeah, so my job is interesting now because I've actually segued out of research and development. So I was always a hardcore research and development scientist, obviously tinkering with like cloning molecular constructs to then in vitro in vivo studies. Well, that was a whole thing during my PhD. But then at Quell, I was doing both R&D, and I kind of did bits and bobs of something called like product development, which was segueing into like process development. Now in biotech companies, and I think in pharma as well, I just don't have much experience in pharma because obviously this is like, tens of thousands of people, whereas in my company it's just like 140, I think. So you have the R&D department where they come up with sort of the pipeline work and the initial proof of concept. Can this work in a Petri dish? Can this work in a mouse model? If it does, great. Then they transfer that to process development team. And assuming that the company nominates that as like, this is going to be the clinical candidate, this is the drug that we want to take into a patient, we have hopes for this. So that tech gets transferred to process development, where essentially they take what the guys in research did, which is, it works in a mouse, but we have to be able to make this in a patient, which is very different. All these other rules you have to follow, because what you can inject in a mouse or like put in a Petri dish is completely different from even the reagents you use, for example. So to the manufacturing process. So process development kind of creates that drug and creates the manufacturing process as best as possible. And then they tech transfer that to the manufacturing and quality control teams. And I sort of sit within quality control, but I'm the lead scientist there essentially. So what I do is I oversee a lot of when the associates are executing the product, the patient lot release. Then if things go wrong, I kind of help do the investigations essentially, and I rope in the manufacturing lot, process development, other people, and we kind of put our heads together to think what could have gone wrong. I'm more of like a technical advisor, I guess you could say, and that's where my immunology background comes in. I realized I jumped a few steps. So with obviously when that comes into manufacturing, we have to make it. So manufacturing team make the product for the patient. So essentially what happens is we'll get a call up from the clinic to say, we have a cancer patient, pancreatic cancer patient, we need to develop this product. And then using what process development gave to manufacturing, like basically the exact protocol, we make it on a large scale in manufacturing. And then what manufacturing does is they hand over during the process samples while it's being manufactured to the quality control team. They just do little like tests to essentially check all is groovy, no red flags. And then at the very end, once we get to the final end and we've made the product, quality control has to do all of these tests as classic QC to basically ensure that we are compliant, what we've told the FDA that the drug, like what it is, its identity, how strong it is, how potent it is, if it's sterile, if it's safe. We had all of these like check boxes that we've rigorously went through with the FDA, and we have to tick those off. And if everything's ticked off, it can literally get shipped out to the patient. So, and then it can go into the patient as the drug. So that's kind of where I sit. I'm not in the lab anymore. I'm not executing any of this, but I kind of help oversee a lot of that. And my immunology background is extremely useful here, because when things go wrong, I can also look at, okay, is something happening in the cell biology context? And it's not even when things go wrong. I don't want to give the impression that things go wrong all the time. I mean, when like phase one, you're always like learning. So we're in phase one at the moment, but it's also like when you're actually bringing a product from research and development to process development, ultimately process development have to make it suitable for manufacturing quality control. So I give my input there, like, well, this is what we want in the experiments that we do to test if the product's safe or the product's potent. This is what ideally we want to put and manufacture it. How best can we play around with the cell biology, just knowing all this information about immunology, and ultimately we are manufacturing immune cells. So I'm in the CAR T cell therapy space at the moment. That's sort of what I was talking about. It's pretty much the same tech I used for my PhD, but for the autoimmune organ transplant rejection space. So essentially what we're doing now instead is we have patients with various cancers such as pancreatic, ovarian, non-small cell lung, and colorectal, or in the UK, bowel cancer. And assuming that they test positive for the certain markers we're looking for, then we extract out their immune cells, and then we engineer their immune cells, specifically their T cells. We engineer their T cells to be able to hunt down, for example, the pancreatic cancer. and then we expand them to like the billions in manufacturing, and then we dose the patient. So now you have these T cells that are armed with this receptor to be able to drive them straight to the tumors. So that's what I'm doing. It's incredible. It's been a game changer. I mean, immunotherapy is just, I'm very biased towards it, but I think it's definitely going to be changing how we treat even other disease types as well that we didn't typically, as I say, classify as immune mediated. So yeah, that's kind of what I do. And it's a bit of a wishy-washy response, but I love it. It's enjoyable. Happy to also be out of research and development, but to still get to attend their meetings every week and still get to have my say and give my input where necessary.
That's amazing. I think you shared recently too, one of the cells attacking, or the T cell attacking the cancer cell on your Instagram, didn't you?
Yes, yeah. So that's not from my like lab, although there was one of the papers that my people in my company published a few years ago, and I did share that video because I thought it was really cool. But I think imaging work like that just captivates people more and makes people interested more because you can actually see the cells in action. So yeah, that example was like a T cell, and there's a couple of T cell examples I've shown where just this tiny little thing is able to absolutely obliterate this ginormous cancer cell, which is just amazing. The various weapons, and that's the other really cool thing, all your immune cells have different tools in their arsenal to be able to deal with threats and use different ones accordingly. And in that particular example, at least in that video I posted, I think it was yesterday, it's a particular mechanism where T cells will basically come and like poke holes in the cancer cell and then just drop essentially the equivalent of like a bomb inside of it, just explode the cancer cell. So that's called perforin granzyme. And I mean, some biologists might be like, oh, she used colorful language. I definitely have. But the perforin is what the little holes that are poked, and the granzyme, those are basically like digestive enzymes. And so they just insert the digestive enzymes in the holes into the cancer cells, and it just, as graphic as you can imagine, just dissolves and kills this evil cancer cell.
Crazy. Like, yeah, when I watched that, I kind of got the Magic School Bus vibes. Is that a Canadian reference? Do you know what Magic School Bus is?
No, I don't know if that was in the UK at all, but
Like in elementary school, it was a TV show where a teacher, Miss Frizzle, had her magic school bus, and she would teach the kids science. And the magic school bus would shrink down and go into a human, and you would learn about the biology, or go into a plant and learn about the plant. It was cool. So looking at your Instagram, I kind of get those magic school bus vibes because you break it down so in a neat way. Now, I know we touched a little bit on the immune system, but for those people who may not have like a science degree or didn't even take like high school biology, because that was a long time ago for a lot of us, like what does your immune system do? Like if you were to break it down real quick, what does your immune system do?
yeah, so your immune system is basically there to protect you when your initial barriers get compromised in some sort of way. So you have all these other barriers in place to stop the world from getting in, because your body is like a temple, it's paradise. Just remember that anytime like you look down on your body, like, oh, whatever, let's get to the gym. Like your body is paradise for all the nasty bugs that are out there. It's warm, it's cozy, it's got lots of nutrients inside. Like everything is just basically trying to get inside. So that's why we've also evolved to have all these barrier mechanisms, like our skin, for example, which is very, very tough and it's got many layers and it's not that easy to penetrate. Even all your potential openings to the outside world, your mouth, your nose, everything has got all these mechanisms in place to be able to stop any pathogen that's trying to get in its tracks, essentially. But as we know, things happen. We get cuts, we get exposed all the time to various bugs and all the stuff that's going around at the moment. So that's when your immune system kicks in. So your immune system job is, well, one of its jobs is to protect you from what could be coming in and just to eliminate it as quickly and as efficiently as possible. So you have your first line, which is we call your innate immune cells, and your innate immune cells are your first responders. So they're the first ones there at the scene. And you have these cells that are constantly patrolling as well, looking for potential invaders, essentially, but also your non-immune cells. So at least in the context of like, I think IBD is a good example. Your intestinal epithelial cells. So I guess one way to call that is your gut lining cells. They also have hallmarks of the immune system where they can respond as well. And they have a very active role with communicating with your immune system. So that's what's amazing is constant communication between your body cells and your immune system helps keep everything in check and in balance. So anyways, let's say you have this invader that comes in, this virus or bug, then your innate immune system kicks in and it can efficiently, quickly clear the infection, or it might call in for backup, which is then your adaptive immune system. So when you think of your T cells and your B cells, and for people that don't know what B cells are, they are the guys that make the antibodies. So there's been so much hype around antibodies the last 4 years, but people forget the guys that make them are the B cells. So the T cells and the B cells, they are more specialized, like evolutionary in the process. They are the ones that come in and they have the ability to retain memory as well. There is some interesting data to support that the innate immune system also has memory, but it's a different type of memory. And while the innate immune system responds to just general molecular patterns that could be associated with a pathogen. For example, just generic bacterial DNA, generic viral DNA. So those are just a couple of examples of many, these conserved patterns that it will recognize. Your adaptive immune system will recognize with such specificity the exact blueprint of what has come in. And that is why it has this memory to that blueprint. Should you get exposed to that particular insult again, your adaptive immune system can kick in immediately. So they all work to clear the infection as quick as possible. And then that's when you have inflammation going on, because then everyone gets involved. Your body cells are involved. Your body cells as well are sort of like they adapt to be able to allow immune cells to come into the area. And then the immune cells call up other immune cells. There's this constant battle and fight scene that is occurring. And that's when you get the symptoms of inflammation, as I say. But then once you've cleared the infection, that's when the cells that I'm biased towards, at least in my past, the Tregs come and they basically shut down everything. Like, all right, coast is clear, bad guy is gone, clear, inflammation, stop. But then the repair and healing process begins. And a lot of people forget that your immune system actually has a role in the repair process as well. So you can kind of think about it as like it's this war zone, but then immediately into construction workers. So, which is really cool. So that's essentially how your immune system works. And it's constantly patrolling. It's even constantly looking for potential cancer cells in your body as well, or cells that have the potential to turn into cancer. So we call this immune surveillance. So they'll go and eliminate any potential threat that could be there. So they're constantly on the move. And they're just constantly trying to keep you alive every single day.
That was lovely. Just like the bit. And then Dr. Yaz, can you tell us a little bit too, when someone is in an IBD flare, what the heck's going on with the immune system?
Yeah, IBD is a very interesting one that we're still kind of trying to make sense of as sort of in the immunology space. So, of course, I caveat with saying that I'm not actively in that research, but I have been reading about it and I kind of know as much as I can really about how the immune system works here. It's complicated because there are definitely genetic elements involved as well here. You know, people with IBD will be genetically predisposed somehow. Now, it doesn't mean that everyone with the genes that are associated with IBD will go on to become full on having IBD. There are usually various drivers of that. And I think right now we haven't identified, there's no real single driver, if that makes sense. Although there's very interesting stuff on norovirus that, for example, has been linked. And you can understand why, at least with viruses, we have seen links to certain autoimmune diseases, and EBV is a massive one. But anyways, moving on. So when you have IBD, what is happening is there's a few things going on. First of all, your intestinal epithelial cells, so that's your gut barrier lining cells, they are sometimes acting inappropriately to maybe antigens that come in, your gut microbiota as well, and then they are stimulating your innate immune cells, such as your dendritic cells, so your antigen presenting cells. So they are the main guys that patrol, and then they have the ability not only to, as I say, capture and clear an infection, but then to call up the adaptive immune system. So we've seen various gene aberrations to do with like these core receptors that your intestinal epithelial cells have, your antigen presenting cells have, which maybe you guys have heard of, like the nod-like receptors and like toll-like receptors. So these are the receptors that are in charge of basically sampling and picking up whether you have a generic pathogen. So they can pick up the generic, like bacterial DNA, as I mentioned, the generic viral DNA. So we've seen mutations in those particular receptors. The receptors are supposed to be sampling and being able to check, yeah, no, this is bad, no, this is good. So what happens is if there's a mutation there, then it can trigger your immune cells as well to then behave inappropriately to something that should be a friend. And then what happens is this sort of perpetuates the cycle. So if you have the dendritic cells, which are the guys that link the innate and adaptive immune system together, acting inappropriately, then they'll recruit the adaptive immune cells as well. And I know with ulcerative colitis, they call it mainly a T helper 2 response. So that's mainly that's your T cells can be different arms of T helper 1, T helper 2, T helper 17. And these hit in the CD4 T helper cell category, which are different to the cytotoxic killer CD8 T cells. With Crohn's, we mainly see it as a T helper 1, and ulcerative colitis, we mainly see it as a T helper 2. But we do have other, like the T helper 17s get involved. And basically, these T cells coordinate with your B cells to make antibodies. They coordinate with your innate immune cells, other innate immune cells, your neutrophils, and other phagocytes come along. And the problem is when you have this initial aberrant response that shouldn't be happening, that inflammation, as I mentioned, these immune cells have the ability to recruit a bunch of other immune cells, release all these other mediators, get your non-immune cells involved, and that's when you have that flare. That's when you have the sort of leakiness that occurs as well. So that's why also we have to really get it under control. And that's the part where biologics come in, for example. But that's also why then you get like the lesions and the ulcerations within your gut that you see through colonoscopy as well. It's this inflammation that's going on. It's this damage, constant damage to the gut. So what happens, as we say in autoimmunity, is your immune system is attacking you. For some reason that we call tolerance, so tolerance to self, as in you, gets lost. So in IBD, your immune cells end up attacking various gut antigens. So various like proteins, we can call it various proteins within your gut, and it shouldn't be attacking. So that's why, at least what I do with immunotherapy, we could apply that context to an IBD, where you can take Tregs and engineer them to go localized to the gut, to protect the gut. I know there's a lot of proof of concepts right now out there looking at that. But yeah, I know it's a bit of a waffly answer, but hopefully that makes sense. We obviously are looking at the roles of different immune cells. Oh, and one more thing. Tregs usually are either deficient in number or dysfunctional in some sort of way in autoimmune diseases. That's a key thing I should have said. So we have seen, at least with gene loci as well, like IL-10, which is a major protein released by Tregs. So IL-10 is a cytokine. Every time you see ILs, they mean different cytokines, and cytokines is the language immune cells use to speak to each other. So for example, it could be to recruit other immune cells, to tell immune cell, you need to differentiate to do this, you need to do that, to bring about different mechanisms, different responses. So it's the language. IL-10 is released by Tregs, and its core role is anti-inflammatory. So of course, when you have these Tregs that have issues with the IL-10 gene, that impacts the ability to have this anti-inflammatory state, so that can also contribute to these pro-inflammatory responses that cannot be controlled by your regulatory cells. So there's a lot going on. It's an absolute hodgepodge. And people are interested in how this cell's involved, how that cell's involved, how this gene has some sort of ability to be able to impact that cell, but also there's a bunch of genes that we don't even know what they do. We just know that they're associated with IBD, but we haven't been able to figure out how. Yeah, we're still working it all out. Hopefully that makes sense.
Right. It's really interesting. I never thought of cytokines either as communicators, or understanding in that deep way that they're actually just communicating with other parts of the immune system.
Yeah.
I had a question too. So what about when the immune system just forgets? For example, I'll give you a personal take. So my mom got shingles a year ago, and I was concerned about it, but then, the doctor was like, well, if you've already had chickenpox, you're fine. And I was like, well, let me just double check, because I don't really know if I trust my immune system. So then I did like a check to see if I had antibodies for chickenpox, and I didn't, even though I had had chickenpox earlier. So what's going on when the immune system like forgets stuff like that? If you've been exposed to something, you've fought it off, and then all of a sudden you don't. Your body doesn't remember fighting that.
That's interesting. So whenever you looked specifically, you said that, yeah, you checked for antibody titers. Was this like straight after, like how soon after did you check for like your antibody titers? I'm assuming that's what you looked for.
I had taken it before my mom got it. And then I, my doctor was like, let's run it again. So then, I was around her, taking care of her, all that. And then a couple months later, then I did have antibodies. So it was just this weird scenario where I was like, what is going on? And I don't know that my doctors had answers either as to why I would have had chickenpox, but then not had antibodies to it.
That's interesting. Yeah, I'm not too sure about whether there could be immune amnesia there. I mean, I think so. Basically, that's mainly associated with like measles virus, and I know there's right now there's a scare with measles at the moment. There's some outbreaks in the US at least. I think different parts of the world as well, actually, where there's this immune amnesia can occur, which is very scary, but essentially it has the ability to wipe your pre-existing immune memory. And as we mentioned, those are like your adaptive responses, like your B cells and T cells. So they form this memory that basically go and sit in your lymph nodes, your spleen, your lymphoid organs, but also can sit in your tissues and reactivate where necessary. But I need to double check that. I don't think that that's something that shingles can necessarily do. What I will say with what could be going on is that antibodies don't also tell the full story. So, I mean, I'm quite biased as a T-cell immunologist as well. So when you obviously get exposed to something, or when you have a vaccine, for example, your B cells will be churning out these antibodies. And so the B cells that do that are called plasma cells. So these plasma cells are constantly like producing these antibodies, but then you have short-lived plasma cells and you have long-lived plasma cells, right? So sometimes the antibodies, as we know, like with COVID, for example, they don't last for many, 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 many years, the way like for the MMR vaccine, which you can have these long-lived plasma cells that can continue to churn out antibodies for decades. And, there's multiple reasons for that. But essentially, the type of exposure, the strength of the exposure, the antigen itself, for example. But maybe in your case, at least with shingles, I mean, as I say, I'll have to double check that. I don't think there's any association with immune amnesia, but we can follow up in the show notes to something if there is. But, there's just possible that you have those memory B cells and your memory T cells still there, but there wasn't a need to actively produce antibodies. Like your immune cell also strives to conserve energy as well, and it's efficient in that regard. If antibodies don't need to necessarily be there actively circling because there's no threat, they won't necessarily be there. It doesn't mean that if you don't get exposed again, those memory B cells will kick back into gear and will be producing those antibodies again. So hopefully that helps. And also don't forget about your memory T cells. And T cells don't produce antibodies, but they're very, very powerful. So hopefully that helps a little bit. Yeah, it's very nuanced. It's very complicated. I mean, I know right now there's a lot of panic about, at least with measles, as I say, like, oh, my titers for MMR. And like titers don't always tell the full story. And there's obviously some complications there in the sense of like, if you don't have titers there, then maybe you could get just another dose. But then for those that are on biologics or immunocompromised, you shouldn't get another dose of these of them. I don't know if that helps in any way.
Yeah, I never thought about that. I didn't know that the titers only tell part of the story. That's definitely an interesting way of thinking of it all. But yeah, we'll follow up in the show notes if we find anything else out. And then I guess another question we had was around the role of stress in the immune system. So I know with IBD, we can look at, short-term stress can increase symptoms, and then your longer-term stress, you might see more an increased risk of flare. But what's going on at an immune system level when you have long-term stress at play?
Oh, stress is a very interesting one because cortisol has become such a buzzword on social media, which I find a bit strange. I definitely would like to sort of do a bit of a deep dive on that on social media. I'm very interested in this myself, just from my own situation. There's always aspects that you think, which are toxic, as self-blame, like, oh, was I too high stress with everything going on in my life, and sort of too much of a perfectionist, and maybe I manifested my symptoms. I'm definitely not as serious as that. But stress is, first of all, stress is a good thing on the body, in the sense of your immune cells have these receptors to respond to the hormones that are released by stress. So with acute stress, obviously you have the epinephrine released. You also have cortisol released, and these are really good from an evolutionary standpoint as well. Like initially, like the epinephrine cause the immune cells to mobilize quickly, and to be able to respond to threats. It's really beneficial in that regard. Cortisol has this dual role. People accept that cortisol does have a pro-inflammatory role, but it's more classically defined as having an anti-inflammatory role. So cortisol, so that suppresses things, keeps things in order a little bit more. And this is all good. Different stresses can cause this release, from physical stress to then the mental and emotional stress. Now, slightly different pathways and a little bit more complicated in terms of the actual mechanism. Although they're stressed, they actually act a little differently on the immune cells. Exercise is more beneficial, but then too much exercise can push you too far the other way as well. So there's a lot we could definitely talk about there. But with long-term stress, like chronic stress, there is this sort of thought behind cortisol resistance occurring. So your immune cells maybe aren't necessarily responding appropriately to cortisol. I know there's also new theories being pushed out, as I said, about actually no, it's more cortisol having this more pro-inflammatory role. It depends who you chat to, different labs and stuff in the stress space, because I've been interested in this. But essentially your response becomes a bit more dysregulated. So your immune system doesn't behave appropriately to cortisol like the way it should be when you have chronic stress. So this, general sort of maybe anti-inflammatory response, or in certain situations, pro-inflammatory response, in others, it gets really mixed up and it doesn't behave how it should be in that moment. So that's what can happen long term. And we know this for certain because even when looking at cancer patients and immunotherapy, there's some fascinating studies looking at the role of stress on cancer patients, and even how therapies, how patients respond with their therapies if there's a lot of stress, if they're undergoing a lot of stress. And that's why there's interesting studies looking at beta blockers, for example, like propranolol, to try and see if that can help actually your immune cells out and responding to the therapy, because stress also exhausts your immune cells. And when you exhaust your immune cells, it's not going to be that good in fighting infections and fighting cancer, for example. And then it's more likely to also switch to become inflammatory towards you. That tolerance can be lost. So there's a lot at play here. It's not that simple, of course. I know people like a reductionist view of stress has caused this. It's really not that simple, unfortunately. But we do know that stress definitely contributes and plays a role to these aberrant responses, at least if you have chronic stress. Hopefully that makes sense.
Yeah. And I was reading too that with stress, it also depends on how you interpret the stress too. Like moving might be stressful for one person and might be totally chill for another person, or moving across the country or moving to another place, or like grief, or, there's all these different stresses we can have, and we can have a completely different body response to it. So it's kind of even hard to define what is a stressor, and what is the long-term stressor.
Exactly. And people have different tolerances as well, right? So like different resilience, I guess you could say. So it's just not that. It's not one size fits all, really. But we just know that your immune cells have the receptors, so they do respond. But yeah, it just different people respond to varying degrees.
Wondering, I do have one last question, but I don't know if we have enough time to cover it.
It might be a longer one, because
Is it?
maybe we need a part 2.
One of them, one of the questions was that we talked about pregnancy and IBD quite a bit in past podcasts.
Yeah.
And it was kind of, helping us to understand, like, why do some autoimmune diseases improve during pregnancy and some get worse? And, what role does estrogen play in all of that?
Yeah.
But that could be long.
Yeah, oh my gosh, estrogen. So I did like a couple of posts on this as well. I mean, we can see how far we go. I'm happy to talk about this in a part 2 if you want as well. But, your immune cells do have estrogen and androgen receptors, and I did this post on trans studies, trans studies are fascinating, realistically, where people sit in terms of
You're talking trans, right?
Sorry?
Gender, like trans.
Exactly, yes, gender transitioning, because we know your fixed chromosomes, obviously, men and women have different immune system profiles, and a lot of that is also down to your fixed chromosomes. Most of your immune genes sit on your X chromosome, and that we found studies that basically is this issue where you need X inactivation to occur, because obviously we've got 2 copies, and the idea is that one of them gets inactivated, and that kind of is in line with the men's single X chromosome as well. But then there are issues when there's no X inactivation that has been linked to autoimmune diseases as well directly. It's really, really interesting. But we know that chromosomes just tell part of the story. Hormones play another massive role. And when those that are transitioning to either become trans males, for example, may have more of the take on more of the cis male immune profile. So suddenly more suppressive, not doing as well to viruses, not building as much of a robust vaccine response compared to cis females. Whereas those that are transitioning to females, so trans females, may be at higher risk of autoimmunity. They have more of the type 1 interferon responses, which are like the antiviral responses kicking in, better vaccine responses. But again, this downside of more risk of autoimmunity. So we know that sex hormones play a role for sure. Now, when estrogen comes in, it's very, very interesting, because pregnancy is a whole different beast, because in pregnancy, you are having to do what we said, which is accept a non-self. So the fetus is half you, half someone else, and your immune system can't reject it, right? So that's why, to bring a baby to full term, so your immune system has to remodel to basically allow this half you, half not you grow, develop, flourish, and then become a beautiful bouncing baby at the end. So that is when your Tregs get involved, your other M2 macrophages, all your other regulatory immune cells get involved. Now, that is why a lot of people might see that autoimmune disease is going to remission. But it's not that simple, because your different autoimmune diseases have different, as I mentioned, predominant B cell or T cell responses. So we know with lupus, it's mainly B cell, like antibody driven, for example, but also T helper 2 cells are involved. As I mentioned, with Crohn's, it's more T helper 1. With ulcerative colitis, it's more T helper 2. Now, traditionally, again, more complicated than that, but a lot of immunologists would agree that the T helper 2 sort of errs more on the anti-inflammatory side versus the pro-inflammatory. Again, very nuanced, because, allergies are T helper 2, and that's not anti-inflammatory. It's very pro-inflammatory, but the idea is that your immune system skews to more of this Th2 profile. That's why, for example, in some cases, Th2 predominant autoimmune diseases can get exacerbated, whereas Th1 or Th17 autoimmune diseases can calm down. It's very nuanced in general, though, because it's not all about the T helper cell type. You have other immune cells at play. As we said, the hormones, there's a lot of different stresses going on. So it's not just like a cookie cutter approach. But that is why some people, at least with Crohn's disease, may go into remission or may have a downplay of their symptoms because they're mainly Th1. And then if their immune system remodels to more of a Th2 sort of regulatory, it could calm it down. And again, it's not as simple as that. And there are different phases as well, because during pregnancy you have different levels of hormones at different trimesters, and that also complicates and muddies the waters a little bit. But I mean, we do know that during a menstrual cycle you see immune system changes happening, so it really is just that acute, just basically based on the high estrogen and low estrogen. We could talk about this for ages. I'm very fascinated by this as well for myself. Sorry, my cat is running around. But yeah, that's just a little snapshot. And it's just, I really want to stress, it's really not that simple, though. Because
that could be the title of this episode. It's really not that simple. Yeah.
It's
so fascinating. And I just so appreciate you being here and taking some time to talk and nerd out about the immune system with us. So thank you. And thank you for all that you're doing to educate others too on Instagram and elsewhere. So appreciate you. And hopefully we'll have a part 2 and we can get more in depth about pregnancy and what hormones and stuff.
Yeah, that sounds great. I would love that.
All right. Take care.